Gracias. Y primero quiero decir gracias a doctora Pennington por invitarme a este congreso y es un placer de estar aquí con ustedes. Y también quiero decir lo siento porque es necesario que hable en inglés. Pero si tiene preguntas, con las manos. Bueno, sí, so my talk is going to be a little different from the title that you said. Today, what I'm going to try to talk about is a new type of science, well, relatively new, called genomics. ¿Y hay alguien que conoce genómica? No? Bueno. So good. So you get to learn something today. This is good. So what is genomics? That's hopefully by the end of my talk, I will have helped you understand a little more what this means. And so, and myself, I don't really actually know what it means either. So it's a difficult word to define. So I'm going to try from a few different points of view to try and understand what is genomics. So in many ways, genomics is a new way to study biology, is one idea I had. It's a different way to think about biology. So what do I mean by that? So really need to compare genomics to traditional biology. No. See? So see, with traditional biology, you basically you go out into the field or you go into the laboratory, and step one, you observe the organism. So you look at the organism. And you look at the organism and you think of a hypothesis. And then you go into the laboratory and test the hypothesis. Could I get the handheld mic? This works? See? Okay. Yeah, so then you look at the organism, you test the hypothesis. So that's the scientific method. Bueno. Genomics is very different because step one, you sit down at a computer. So this is the big difference. Step one, you're at a computer. You're nowhere near an organism. There's not an organism in sight. And what do you look at? You observe the genome sequence. So does everyone know what a genome sequence is? The sequence of DNA, see? Sequencia, see, okay. So step two, after looking at the genome sequence, you collect large sets of data using big fancy machines. So you collect thousands and millions of pieces of information. And you're still not done. Then you put that data back into a computer and you stare at the computer screen, and at the very end of the process, kind of one of the long goals of genomics is to make a computer model of how the organism functions. And so, in a lot of ways, one of the goals of genomics is to take the organism and put it into a computer. So to put its whole essence into a computer so you can interact with the organism using your computer. So that's kind of an abstract way to think about genomics, but it gives you an idea that this is a different approach to understanding biology, where we're using a computer as a tool to let us see things that we could not see otherwise. So genomics is, in a lot of ways, is similar to genetics, and it's really a science that grew out of genetics. And so how are those two things different? So in traditional genetics, when we're trying to understand a biological process like flowering, what a scientist who studies genetics will do is take a small number of genes, study mutations in those genes, and build what's called a pathway. And so in this pathway, each of these little names is a different gene, and the arrows connecting them indicate that the genes are interacting with each other. So this is like a little map to understand these small number of genes that control a biological process, in this case flowering. And so a genetic scientist uses mutations like Claudia was telling us about that segregate in progeny, and they look at the organism and the segregation and they build a model. And so that's traditional genetics, like Gregor Mendel used that approach. With genomics, what we try to do is use the power of computers and the power of a database and the power of the technology to collect large data sets so that we, at the same time, analyze all of the genes in the organism. And so you end up with a map like this that shows the relationship from all the, in a human, 30,000 genes, all the lines, how they connect. And when you compare these, again, it becomes really obvious that you need a computer. If you're going to do genomics, you have to use a computer. It's critical. Okay, so I still really haven't said what is genomics. We still haven't actually talked about that. And so one way that I think about genomics is to, I think that it's a science where a complete genome sequence gives you the foundation for doing your experimental biology. And I think that's a pretty good way to think about genomics, because all of the experiments that get done by someone using genomics, the foundation is that whole genome sequence, so the GATCCGATA of every chromosome in the whole nucleus, all those letters. Humans, it's 3 billion? Does anyone know, is that right? Someone knows? Let's say 3 billion, but a very large number of those base pairs. Okay, so this is my silly little picture, but it's to give you kind of a visual appreciation of what I mean by foundation. So if I'm like building up a genome center, like doing a genome project, the first step is to get my computer and load in the complete genome sequence of my organism. And once I have that foundation, then I can start doing experiments where I collect these large data sets that I told you about before, which are really important for genomics. So once I have the whole genome sequences, which is all the DNA, then what I can do is I can start measuring all the RNA, which is transcription, which is also called the transcriptome. So in genomics, a lot of the terminology, you take a word and you just add OME to the end, and then it becomes an ome. So this is the genome, gives you the transcriptome. Once you know all the genes that are being expressed, RNAs turning up, turning down, then you can ask questions about the proteome, which is all the proteins that are floating around in the cell, which are being regulated up and down. And so, you know, one thing to remember about basic molecular biology that you will have learned in your classes is that DNA is the instructions, RNA is the cell reading those instructions on and off, and then the protein is those instructions being turned into an actual molecule that does work in the cell. So the DNA becomes the RNA, becomes the protein, and once you have those proteins floating around in a cell, they carry out metabolic reactions, and you get all these small molecules that are also floating around in the cell, and that's called the metabolome. So the transcriptome, proteome, metabolome, these are those big data sets that you're collecting and putting into your computer. And at this point, you're generating a big mountain, or I guess in this metaphor, a big tower of information. And at this point, it becomes unmanageable. And so that's where bioinformatics and a field of science called systems biology is where people who are experts with computers try to write programs that let the scientist assemble all this information into some meaningful interface where they can spend time understanding it. So you can take all these big data sets and use them to actually understand the organism. So that's just kind of the overview, kind of the introduction I wanted to give you of the relationship between the DNA sequence as the foundation and these different fields of genomic science and how they build upon that foundation. And so I mentioned a little bit earlier that a really important part of genome science is technology. And that's really true of all experimental science. In a lot of ways, we're limited by what we can observe by the technology that we have at our disposal. And so in the case of genomics, each of these levels of the genome tower has their own technology that's really important for letting scientists perform these kind of observations. And so the foundation of genomics is genome sequencing. And so that's just the very simple process that we talked about, where you take all the DNA for a human out of your body, you put it in a machine, and the machine starts reading the chromosome G, A, T, C, A, C. There's only 4 bases, so it sounds really simple. But the trick is to be able to read all 3 billion all in a row really cheaply and really fastly. So in the year 2000, the first human genome sequence was completed. It was a 13-year project, and it cost $3 billion US. So this was a very big project. And this was laboratories all over the world working together as a collaboration. 13 years, $3 billion. In 2007, 7 years later, as engineers were making the sequencing machines cheaper and faster, it cost $1 million, and it took 2 months to sequence 1 genome. 2 years later, there's a company in California called Complete Genomics, and they can sequence 1 human genome for $5,000, and it'll take them 1 week. And so you can see that this technology is rapidly advancing. The easiest comparison would be like your, I don't have my phone here, but your cell phone technology. Think about 10 years ago, what did a cell phone look like? I don't know, it was like this big, probably bigger than this microphone. And now you've got your little magic phones where you watch TV and listen to music. The same technology has been increasing in DNA sequencing. And that's all being driven by the industry of medical genetics. And so just as a comparison, there's a category of sequencing machines that are called next generation. And they're kind of a big deal in the genomic field. The first human genome that cost $3 billion was done on a machine that could generate 96,000 bases per run. So in one day, you put some DNA into the machine, it could tell you 96,000 letters, which is pretty good at the time. That seemed pretty great. Until these guys came along and made this other machine that does 250 billion. So this machine is 2 million times faster than the other one. So that you can imagine, that lets you generate data 2 million times faster. So again, we get back to the idea that we really need computers in order to do genomic science. The other thing, that's really important to understand is this science is not just something that lives in the laboratory at the university. Genomic science is something that is extending out into the lives of regular people all over the world. And if you look on the internet now, there's a project called the Personal Genome Project, where they're trying to enroll 100,000 participants from the general public who will donate some of their DNA to the scientists, and they will read all 3 billion letters in your genome. And this project is something 10 years ago, if you told someone you wanted to do this project, they would have thought you were crazy. Because, you know, it costs $3 billion to sequence the genome. How are you going to do 100,000? 5 years from now, it's going to cost $200 to sequence the genome. So this kind of project is going to become very, very doable in the near future. And so not only are there academic projects like this, there's also consumer projects. There's a company that's on the internet called 23andMe. Can anyone guess what the 23 means? Very good. So it's a company where let's see, they send you a little kit, and the kit has a little stick in it. Can anyone guess what you do with the stick? Yeah, you scrape it around in your mouth, and you put it in the package and you send it back to them, and the company will map a bunch of mutations in your genome, and they'll send you a little report that tells you, you know, I have my bias, it's really stupid. They'll tell you like how smart you are and you know, where your ancestors came from, and if you have hairy ears. It's like, well, you can just look in the mirror to see if you have hairy ears, but it's a way to make money. And they had a holiday sale in December where instead of $500, you could get your genome done for only $99. So anyway, this is just to show you that this genomic science is starting to reach out into the lives of regular people who have $99 more than they need, I guess. So where else is it growing? So there's these companies doing these projects. In China, in Beijing, there's this new research center that's the biggest DNA sequencing center in the world. And they've started a project they have running now where they're selecting 1,000 different organisms from the whole tree of life, like fish and alligators and trees and bugs, and they're sequencing the whole genomes of all these organisms. And they're also doing 10,000 microbial species. And how can they do this? Remember I told you earlier about that machine that does 250 billion base pairs a day. Well, they have 128 of those machines, and they have 3,500 scientists whose only job it is to read the letters GATC in any organism they can find. And so they can sequence 11,000 genomes in a year. So again, this is just to show you that this science it's industrial, it's large scale, it's growing, people are throwing money at it. But I'm showing you this slide just to remind you that unlike the people at 23andMe who want you to believe that if they read your genome sequence, it's a crystal ball and they can tell you all about your life. Your DNA isn't your destiny. So it's the old biological question, nature versus nurture. And you know, the answers, of course, they're both important. And so not only is it the DNA inside the organism, but it's all the influences from the environment that determine what the final phenotype is, what the final behavior is. So nothing is ever as simple as people make out. Okay, so that's the technology that lays the foundation. It fills the database with whole genome sequences. You know, it started out in the year 2000. People were excited because there was one human genome sequence. Now, people are only going to be excited when there's 100,000 individuals. So we've got that foundation. But like I said, the genome sequence is just the instructions. It's the blueprint for the cell. The transcriptome, which is the RNA, that tells you what genes are being switched on and off. So measuring if a gene is on or off. Of the 30,000 genes in my body, which ones are being switched on right now and what ones are switched off. That's what the transcriptome is about. And so there's a technology called the Dna Microarray. Who's heard of that? People heard of microarray? Yeah, so they're tiny little spots of DNA corresponding to different segments of the genome. There's a company called Affymetrix, which is the main company that manufactures these chips. They're from the same industry that makes computer chips, so it's standardized, miniaturized fabrication. And they have 800,000 different DNA spots on these chips. And so that lets you measure very precisely by looking at the intensity of the light that's coming off each spot, whether a gene is switched on or switched off. So the gene at this spot is turned on. This dark patch here, whatever gene sequences at that spot is switched off in that sample. So what you can do is scientists all over the world, they buy these gene chips, these microarrays from Affymetrix, and if I run an experiment in my lab, I collect data on some plants. I'm a plant scientist, so I grow some plants under different conditions, and I measure the gene expression of the whole genome. I put that data into a database, and now I did that experiment in my lab, but I want to share that with people all over the world. There's this really amazing database that if you write down this web address, tonight when you go home, if you're interested, you can log on to that website for free. And you can analyze the same transcriptome data that scientists all over the world are analyzing. And today, or when I made this slide, there was almost 55,000 microarray experiments had been deposited into this database. So there's 55,000 experiments that you could access and analyze to study a particular gene or process you're interested in. So this is one of the big powers of genomics, is that it lets scientists share their raw data with each other in a standardized format. And again, that brings in the importance of computers, and it brings in the importance of the internet in genomic science. And one thing I should just point out, these fancy sequencing machines I told you about that are used for figuring out the genome sequence, those same machines have now started to take over as a way for replacing microarrays. So 10 years from now, microarrays probably will not be used. Everyone will just use DNA sequencing to measure all the expression. Okay, so we've looked at the genome with the DNA sequencing machine. We've looked at the transcriptome with the microarray. The next step in the tower of genomics is the mass spectrometer and the proteome. So measuring the proteome is much more challenging than measuring DNA or RNA, because proteins are chemically much more variable than DNA or RNA is, because DNA and RNA, they're just 4 letters, G, A, T, and C, all in a row. Chemically very monotonous, very easy to process. Proteins, on the other hand, there's 20 amino acids. Some are hydrophilic, some are hydrophobic, some are charged, some are neutral. When you mix those together in a protein, it becomes very challenging to analyze. And so the science of proteomics is much more difficult. But the basic idea is you use a machine called a mass spec to measure the level and abundance of all the different proteins. It's the same idea, catalog what's present. And so why would I need to study proteins? And one reason is that the abundance of a protein doesn't necessarily correlate with the abundance of RNA. So I could see a gene expression go way up on the microarray, but the amount of protein doesn't go up. And so it's kind of a technical issue, but it's important to directly measure proteins. Okay, so that's kind of a summary now, just to review. We've got the tower, the different levels, and at the top, systems biology. So now I just want to talk a little more about what exactly we mean by this term systems biology. So step one, you make a list of all the genes, proteins, and metabolites. So that's again using these technologies. Use the DNA sequencer and the microarrays to list all the genes, proteins. You measure the levels using the microarrays. You repeat the measurements under a whole range of conditions, and then you make a computer model. And so as I mentioned at the very beginning, kind of the long-term goal of systems biology is to come up with a computer program that simulates how an organism behaves. So the idea is you could sit at a computer and let's say it has a model of the human, and let's say you're a doctor. A patient walks into your clinic and they have a certain set of symptoms, like, they have red spots on their face and their right arm hurts. So you type that into the computer and it could explain, okay, here's the possibilities of what could be wrong with this patient. And then you can say, okay, well, ask the computer, what if I give the patient this drug? What will happen? And the computer would predict how the patient would react to that drug. So it's a predictive model. And so that's the key word, I guess, is it's a predictive model of the organism. Okay, so now I have this one final thought, which is good because I only have 5 minutes, so actually 4 minutes now. So, I've been using the word computer a lot over and over again. So I'm going to claim that genomics is changing biology from what we've traditionally thought of as experimental science into what could really be called an information science, meaning that you need computers to manage all this crazy information and boil it down to something that the human can still interact with. Because the problem with humans is we build all these machines that let us collect so much information that all of a sudden we don't know what to do with it. We've got this big pile of information. He's like, wow, what do I do now? So that's why we need the computer. So here's just a fun little story to leave you with. Sometime in the 12th century, someone somewhere, I'm guessing in the Middle East, there was a doctor, and they were treating a patient, and they made some observations, and they thought, I should write this down so I can share this information with other doctors in the area. So they wrote down the story about how they had a patient come in with a headache, and they gave them a treatment, and this is what happened. So this is a real document from the National Library of Medicine. So now fast forward to the year 2007, here's a journal article from a medical journal where a doctor has given some patients different doses of aspirin and observed how that affects their cardiovascular disease. So in both of these situations, the scientist interacts with the subject, in this case a patient, does some treatments, takes notes, thinks about it, and then they go to their computer and they write a story. Or in this case, they go to their tablet and they write a story. So you're taking your experimental data and you're filtering it through your language and you're writing down a story. It's story time. So you create a narrative that kind of summarizes what you've done, but it filters it because it's going through the human brain and it's adjusting it so that it becomes a story. So really, in a lot of ways, nothing's really changed in 900 years the way scientists interact with their data and how they share their data until now. So genomics, it's not just changing the way we generate information where we have these big computers, these big machines generating information. It changes the way we share information. And I think that's very important because right now, if I do an experiment in my laboratory using a gene chip, I don't take that information, look at it with my eyes, reduce it down to a table in a paper, and write a little story in English language about what I observe. I actually publish the raw data. I put my complete raw data into a database so scientists all over the world can interact with my raw data and think about it from their own perspective. And so that's really a really big change that's happening in science, where we now have a global sharing of raw data that was not possible in the past. And it only makes sense in the context of genomics, where you have standardized methods, where you have uniform ways of collecting data. And so I think that's an important change. And so this is just, getting back to this thing with the microarrays. This is one example. There's many different types of these websites where people share their raw data with other scientists around the world. So there you go. That's all I have to say. Thank you.